As you know from the previous video that carbohydrate digestion and that carbohydrates are actually in the form of amylose and amylopectin. So as we have discussed that these two amylose and amylopectin, it is nothing but the starch molecule. So almost all starch can be digested inside the stomach, and some starch which is remain as such, that is known as resistant starch, and that can be only digested inside the large intestine in presence of E. coli bacteria. Therefore, we have seen the E. coli bacteria is also very important. If you have not seen the video, I'll post that in the I link button. Please go and check that part of starch digestion. Today we are going to discuss absorption of carbohydrate. Hello students, welcome to medicoRAS. Absorption of carbohydrate takes place inside the enterocytes. So this enterocyte cell has a special structure which is nothing but known as SGLT. So as you can see here, this structure let us consider as SGLT, and this is known as sodium glucose link transporter. So this sodium glucose link transporter, by seeing the name only, one can say that this is nothing but the co-transporter. So as we have used some kind of transporter here, so this is nothing but the protein molecule, and this kind of absorption is known as fascilated diffusion. Therefore, it is very much clear that is, in case of absorption of carbohydrate molecule, we need to go for fascilated diffusion. Now, here we have sodium ion inside the lumen of the intestine. At the same time, we have multiple numbers of glucose molecules. So, this one glucose molecule can enter into the enterocyte cell in presence of 2 sodium ion. So we can say that 2 sodium ion is responsible to carry 1 glucose molecule. Please note it down somewhere so that you can easily understand that what is the fascilated diffusion going on inside the lumen of the intestine. Now let's check how the further absorption will be taken place. As you know that below the enterocyte cells we have blood vessel, so we can represent the blood vessel in this manner. So here we have drawn the blood vessel, and this structure is known as enterocytes. You can see here we have already entered 2 sodium ion and glucose molecule. Now as you can notice that the glucose molecule and the sodium ion present inside this, therefore we have to remove this glucose molecule from that particular zone with the help of another protein molecule or the channel that is known as GLUT-2. So GLUT-2 is exactly stands for glucose transporter 2. So through this glucose transporter, glucose molecule can easily be absorbed inside the blood vessel, and we have now multiple numbers of glucose molecule, which is again going to convert it into energy with the mechanism of cellular respiration. But the sodium ion is also need to remove from this particular zone. For that, another special kinds of pump we have inside the same structure. So let me draw the structure of GLUT-2 somewhere like this. Along with this GLUT-2 pump, we have another pump inside the cell, and that is nothing but known as sodium potassium pump. So as sodium ion present inside this cell, it has to remove from the enterocytes and it has to enter into the blood vessel like this. Again, the potassium ion goes inside this enterocyte cell. Let me tell you one important information here, like wherever we have sodium ion, then automatically water is also going to absorb by the simple diffusion. So likewise, we can absorb glucose molecule, at the same time we can absorb sodium ion molecule and water molecule. So this is the simple way of absorption of glucose. So this absorption process is actually for the glucose molecule, for sodium molecule, at the same time water molecule. I hope it is pretty much clear to everyone that how we can absorb glucose molecule. For next upcoming video, keep tuning and do subscribe. Thank you so much.